Chapter 2, Biological Molecules. Introduction. All material things in this world are made of chemical elements. These elements are composed of atoms. The atoms are joined together forming molecules and compounds. The bodies of living organisms are made of some of these chemical elements. The most common elements in the bodies of living organisms are carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. Of these, carbon, hydrogen, and oxygen constitute about 95% of human body by weight. Other elements are present in small proportion. Biochemistry is the science of biology that deals with the structure, composition, and function of molecules that are present in the bodies of living organisms and chemical processes occurring in them. The bonding of hydrogen, oxygen, nitrogen, and other atoms to carbon atom forms a compound called organic compounds. The organic molecules play a most important role that characterize the structure and function of cell organelles. Carbon-carbon bonds make chain of carbon atoms which form the skeleton or backbone of large organic molecules. The chemical analysis of protoplasm shows that it is composed of two types of compounds. For example, organic compounds and inorganic compounds. These compounds are present in somewhat different proportion in different organisms and even in different types of cells of the same organism. Water is the most abundant of all compounds of protoplasm. It forms almost three-fourths of the body. Water are proteins. Proteins are the most abundant compound present in the cell. Proteins have structural, functional roles in the cell. Other organic compounds include carbohydrates, lipids, and nucleic acids. Enzymes, which are proteins in nature, are also present. Hormones, which play a very important role, and inorganic salts are present in small proportions. A typical cell shows the following approximate percent composition of different compounds present inside it. Fundamental biological molecules. There are four types of fundamental biological molecules present in protoplasm. These are carbohydrates, proteins, lipids, and nucleic acids. These molecules are distinguished from one another on the basis of their chemical structures, composition, and functions. The organic molecules and living things such as fatty acids, amino acids, and carbohydrates have characteristics functional groups. These functional groups add to the diversity of organic molecules. Some functional groups are hydroxyl alcohol group, carboxyl acid group, aldehyde group, amine group, amino group, etc. Inorganic compounds such as water and inorganic salts are also present in protoplasm. Water is vital for the life of living organisms. Without sufficient quantity of water, the protoplasm is unable to perform normal functions. True, though inorganic salts are present in small quantities, but they are important for most biochemical functions. Condensation. Large organic molecules are called macromolecules or polymers. These are formed of particular types of small organic molecules or subunits called monomers. Two monomers join together when hydroxyl group is removed from one monomer and a hydrogen is removed from another. This is called condensation. Reaction dehydration. In the process, water molecule is removed. Amino acid join together to form molecule called a dipeptide. The OH from the carboxyl group of one amino acid combines with a hydrogen atom from the amine group of the other amino acid to produce water. in the equation below. Hydrolysis, water splitting. Polymers macromolecules are broken down into monomers by a process called hydrolysis. It is the reverse of condensation. In this process, hydroxyl group from water attaches to one monomer and a hydrogen attaches to the other. This is called hydrolysis reaction because water is used to break a bond. Sucrose plus water, glucose plus fructose. Both condensation reaction and hydrolysis need specific enzymes. Biological importance of water. Most important topic for examination. Water is essential for life. There is no existence of life without water. Allah the Almighty has created all living organisms from water. The body's of living organism contains 70% to 90% of water. It is essential for existence of protoplasm because protoplasm cannot survive if its water content is reduced as low as 10%. Because of its immense importance, water is present in sufficient amount in most of places on the earth. The chemical and physical properties of water are so designed that they are absolutely important for the vital process of life. Polar nature of water. Water is polar molecule. The oxygen in the molecule is electronegative, bearing a partial negative charge, and the hydrogen in as electropositive, bearing partial positive charge. Water molecules are bonded to one another through hydrogen bonds. Hydrogen bonds are much weaker than covalent bonds, but they still cause water molecules to remain attached together. Universal solvent. Due to polar nature of water, it dissolves almost all types of polar substances and is therefore regarded as universal solvent. This facilitates chemical reactions both inside and outside the living cell. Water provides the medium for chemical reactions in cell. For example, when sodium fluoride is pulled into water, the electronegative ends of water molecules are attracted to the sodium ions, and the electropositive ends of water molecules are attracted to the fluoride ions. As a result, the sodium and fluoride ions separate and dissolve in water. Water dissolves all minerals present in soil which are absorbed by plant roots and transported to other tissues. Cohesive and adhesive force of water. The water molecules remain attached together and do not separate because the hydrogen bonding thus develops cohesive force among them, and therefore water flows freely without breaking apart. Water molecules also adhere stick to surface. It can fill a tubular vessel and still flows so that dissolved molecules are evenly distributed throughout a system. High specific heat and high heat of vaporization. Water has high specific heat. Specific heat is that amount of heat energy required to raise the temperature of 1 gram of water by 1 degree Celsius. That means that water absorbs or releases large quantities of heat energy with little change in temperature. This is why the temperature of water rises and falls more slowly as compared to other liquids. This property helps the organism to maintain body internal temperature from liquid temperature changes. Water also has water as the heat required to convert 1 gram of liquid water into vapors. At its boiling point, high heat of vaporization helps animals and plants to get rid of excess body breath during sweating. and transpiration respectively. The presence of hydrogen bond among the water molecule causes water to remain liquid rather than change to ice or steam. Without hydrogen bond, water would boil at 80 degrees centigrade and would freeze at minus 100 degrees centigrade. In such condition, life for living organisms would become impossible. The ice layer at surface acts as an insulator to prevent water below it protecting aquatic organisms from freezing. Water expands at low temperature. Water has unique property. Expands when temperature falls below 4 degrees centigrade. Water is most heavy at 4 degrees centigrade. Therefore, ice solid is less dense than liquid water, and thus is the reason that ice floats in liquid water. Water body freezes on the surface at low temperature. Ionization of water. Water molecules may ionize into hydrogen ions and hydroxyl ions. Very few molecules out of a very large number may ionize. The presence of ions or normal functioning of enzymes. Carbohydrates. Carbohydrates are polyhydroxy aldehydes or ketones are substances which yield such compounds on hydrolysis. Carbohydrates contain either aldehyde or ketone as functional group attached to one of the carbon atoms. They also contain two or more hydroxyl groups. They are the most abundant organic biomolecules in nature. Carbohydrates are organic compounds that are mainly composed of carbon, hydrogen, and oxygen. The name carbohydrate means they are hydrates of carbon in which hydrogen and oxygen is present in the same ratio as in water, that is 2:1. But now many carbohydrates are known that contain hydrogen and oxygen in different proportions. Examples of carbohydrates are glucose, sucrose, starch, cellulose. Carbohydrates are classified into three groups: monosaccharides, oligosaccharides, and polysaccharides. Monosaccharides. Monosaccharides are simple sugar. They are not hydrolyzed, broken down by the addition of water into more simple units. They are easily soluble in water. They are sweet in taste. They have empirical formula CH2O containing the same ratio of hydrogen and oxygen as in water. For example, 2:1. They contain either aldehyde or keto group. They have carbon backbone. They may contain from 3 to 7 carbon atoms. They have named which end in OSC. Those with 3 carbon atoms are called trioses, with 4 atoms tetroses, and 5 atoms pentoses, and so on. Trioses are the simplest monosaccharide with 3 carbon atoms. Glyceraldehyde, but different structural formula. Such molecules with the same empirical formula but different structure are called isomers. Glucose is aldehyde while fructose is ketone. Therefore, glucose, fructose, and galactose are isomers. They are interconvertible. Ribose and glucose, when put in water from ring structures, monosaccharides are white crystalline powders in stereoisomerism. The atom making isomers are joined up in the same order, but still manage to have different spatial arrangement. Optical isomerism as one form of stereoisomerism. Optical isomers are two compounds which contain the same number of kinds of atoms and bonds. The connectivity between atoms and different special arrangement of the atom, but which have mirror images. Oligosaccharides. Oligosaccharides are hydrolyzed to form breakup from 2 to 10 simple monosaccharide units. The units of monomers are bonded together by glycosidic bonds. The oligosaccharides that are hydrolyzed into two simple units are called disaccharides are the most common oligosaccharide. Common disaccharides are sucrose, lactose, and maltose. Sucrose is present in sugarcane and is hydrolyzed into glucose and fructose. Sucrose plus water, sucrase enzyme, glucose plus fructose plus saccharase. The covalent bond that is formed between two monosaccharide units is called lipophilic bond. Lactose is found in milk that contain lactose and glucose. Lactose plus water hydrolyze lactase glucose plus lactose. Maltose is disaccharide found in fruits. It is composed of two glucose units and is found in our digestive tract as a result of starch digestion. Maltose plus water hydrolyze maltase glucose plus glucose. Polysaccharides. Polysaccharides are polymers of monosaccharide units. Monomers, they are hydrolyzed into more than 10 monomers of glucose units. They are tasteless and are insoluble in water. Examples of polysaccharides are starch, glycogen, cellulose, chitin, etc. Polysaccharides act as macromolecules, polymers for the small carbohydrate units. Green plants prepare glucose during photosynthesis, which is immediately converted into starch. When plant needs glucose, starch is again converted into glucose. Starch is stored in plant cells. Starch is a polymer made of many glucose units bonded together, forming unbranched or branched chain. Glycogen is stored in animal cells. It is also a polymer made of glucose monomers, forming extensively branched chain. Cellulose is another polysaccharide formed of unbranched chain of glucose units. It is building material of green plant cell wall and is probably the most abundant carbohydrate in nature. The bond linking glucose unit as a cellulose molecule are oriented differently from starch and glycogen. The cellulase refers to a group of enzymes which acting together hydrolyze cellulose in human digestive system. This enzyme is not present. Cotton fibers are example of cellulose. Chitin as a polysaccharide, it is found in the exoskeleton of arthropods such as crabs and insects. Chitin is a polysaccharide. It is found in the exoskeleton of arthropods is crab and insects. It is also a polymer of glucose, but an amino acid is attached to each molecule like cellulose. Chitin is not digestible. Function of carbohydrates. Carbohydrates perform various important functions. Source of energy. Carbohydrates are used as source of energy. The CH bond and the carbohydrate molecules are broken down during respiration and store energy, and these bonds are reduced, which is most available to the cells for performing various functions. Human blood contains 100 milligram of glucose per 100 ml of blood. Storage molecules. Carbohydrates are stored in the cells as reserve food. Grapes contain as much as 27% glucose. Honey contains large amount of glucose and fructose. Some of these polysaccharide starch and glycogen. Excess amount of food is stored for future use. The shape of molecules affects lots of things besides its smell. Our bodies, for example, can only use right-handed sugar, left-handed sugar as indigestible amino acid. The building blocks of proteins are almost all left-handed. Our bodies cannot manufacture proteins out of the right-handed version. The cell walls of bacteria are one exception. They contain right-handed amino acids. Structural building materials. Cellulose, a complex carbohydrate, as the major structural component of cell wall of green plants. Similarly, chitin is another complex carbohydrate as the structural component of exoskeleton of arthropods. Most important topic, proteins. Proteins are most abundant organic compounds of the cell. They play most important role in cellular functions. They contain elements carbon, hydrogen, oxygen, and nitrogen. Some proteins also contain sulfur. Proteins are macromolecules, polymers formed of units, monomers called amino acids. A large number of amino acids are known. Of these, only 20 different types of amino acid combine in different number and different sequence, forming hundreds and thousands of different types of protein molecules. Structure of amino acids. Amino acids are carboxylic acid having amino group. Each amino acid has central carbon atom called alpha carbon. There are four different groups attached to the alpha carbon. These are amino group, carboxyl group, hydrogen of alpha carbon, and R group. The former three groups attached to the alpha carbon are constant members and are present in all amino acids, while the fourth one, R group, is variable. It is either hydrogen or alkyl group. Due to this variable, the amino acids are different from one another. A bond called peptide bond link amino acid and protein molecules to each other. The peptide bond is found between an amino group of one amino acid and carboxyl group of another amino acid. Thus, dehydration or condensation reaction in which one water molecule is formed. A chain containing three amino acids and two peptide bonds is known as dipeptide chain. A chain with four amino acids and three peptide bonds is called tripeptide, and a chain many amino acids and many peptide bonds is called polypeptide. Most proteins are usually formed of two or many polypeptide chains, hemoglobin and insulin. Hemoglobin is an oxygen-carrying protein in the red blood cells, which consists of four polypeptide chains, while an insulin molecule is consists of two molecules. is highly specific for its normal function. If an amino acid is not occupying a specific position in a protein function, for example, if one out of 574 amino acid and a hemoglobin molecule is not present in a specific position, then hemoglobin changes its normal globular shape and becomes sickle shape. As a result, the disc-shaped red blood cells also become sickle-shaped. In sickle cell hemoglobin molecule, glutamic acid is replaced by valine. Such that hemoglobin cannot perform its function, and a person with sickle cell hemoglobin dies. The size of protein molecules depend upon the number and kinds of total amino acid present in the molecule. Sickle cell anemia is a blood disorder that affects hemoglobin. Sickle cell anemia occurs when a person inherits two abnormal genes, one from each parent, that cause their RBCs to change shape instead of being flexible and disc-shaped. These cells are most stiff and curved in the shape of old farm tool known as sickle. That where the disease gets its name. The shape is similar to a crescent moon. Red blood cells with normal hemoglobin move easily through bloodstream, delivering oxygen to all of the cells of the body. Normal RBCs are shaped like discs or donuts, with the center partially scooped out and are soft and flexible. They can easily squeeze through even very small blood vessels. Shape of proteins. In regard, the shape protein are classified into fibrous proteins and globular proteins. Fibrous proteins. The molecules of fibrous proteins are composed of one or more polypeptide chains which are linearly arranged in the form of fibers. They are water insoluble. Some of these may form sheet-like structures. Examples of fibrous proteins are keratin, found in hair, nails, fur, outer skin, myosin, present in muscle cells. Collagen, which is the most abundant protein in high vertebrates, found in skin, ligaments, tendons, bones, and in the cornea of the eyes. Globular proteins. Globular proteins, as the name indicates, are globular or spherical in shape due to folding of polypeptide chains. They are usually water soluble. Examples of globular proteins are hemoglobin, albumin of egg, white, enzymes, antibodies, and the proteins of cell membranes. Levels of structure organization. There are four levels of organization of protein molecules. This is because each type of peptide chain bends, folds, and twists in particular way within protein molecules. This gives different types. Primary structure as amino acid become spirally coiled. The structure is called secondary structure of protein. When the secondary structure of protein is arranged into a three-dimensional structure, it is called tertiary structure. When two or more peptide chains are arranged into a large size molecule, it is called a quaternary structure. For example, hemoglobin. Function of protein. Proteins perform the most important function in the life of living organism. Proteins are the structural and building materials of cellular membranes called lipoprotein membranes. All enzymes are proteins that speed up biochemical reactions inside the body of living organisms. The digestive enzymes are important for process of digestion. Without their presence, food cannot be digested. Some hormones, such as insulin, are proteins which regulate biochemical processes. Myosin and actin fibers play an important role in the contraction of muscles and movements. Hemoglobin, oxygen-carrying protein of red blood cells, and animals' protein form more structures such as skin, nail, hair, claws, hose, etc. And plant proteins are stored in most seeds for the future need of embryos, for example, beans, seeds, wheat, etc. Lipids. Lipids are a subgroup of different types of organic compounds. They contain carbon, hydrogen, and oxygen. Other elements such as nitrogen and phosphorus may also be present in lipids. Most lipids Most lipids are nonpolar and are insoluble in water, hydrophobic. They are carbon hydrogen bonds and less oxygen as compared to carbohydrates. Lipids are classified lipids, waxes, terpenoids, etc. Glycerols are lipids which are composed of glycerol and fatty acids. The most common acyl are triglycerides containing one glycerol molecule and three fatty acids. Glycerol is a three-carbon compound. To each carbon, a hydroxyl group is attached. Hydroxyl groups are polar and therefore glycerol is soluble in water. The acid portion of three fatty acids reacts with three hydrogen groups of the glycerol so that a triglyceride and three water molecules are formed. This reaction is condensation. The triglyceride molecule can be hydrolyzed to its components. For example, glycerol and two fatty acids. Triglycerides are stored in animals with saturated fatty acids such as palmitic acid are called fats and are solid at room temperature. Saturated fatty acids are stored in animals as fats. Unsaturated fatty acids have one or more double bonds between some carbon atoms. In such molecule, the number of hydrogen is less than 2 per carbon atom. Any more hydrogen can be added to these molecules. Plant seeds, acylglycerol with unsaturated fatty acids are usually liquid at room temperature. Ghee with saturated fatty acid is prepared from vegetable oil by passing hydrogen through it. Intake of ghee should be minimized, as ghee with saturated fatty acid is prepared from vegetable oil by passing hydrogen through it. Intake of ghee should be minimized, as it may store in blood vessels, reducing their flow capacity and increasing risk. The glycerides have high value and usually yield twice as much energy per gram as that of carbohydrate. Phospholipids. Phospholipids are composed of one glycerol molecule, two fatty acids, and one phosphoric acid molecule, usually linked to form to some hydrogen nitrogen groups. A triglyceride molecule is converted into phospholipid when a fatty acid is replaced by one phosphate. A phospholipid molecule. Phosphate head it is polar and is therefore soluble in water or hydrowater philic level. And two hydrocarbon tails they are nonpolar and are insoluble in water or hydrophobic. First find themselves in a double layer in the presence of water in the plasma membrane of the cells. Waxes. Waxes are formed by long chain fatty acids bonded to long chain alcohol. They are solid at normal temperature because they have a high melting point. They are hydrophobic. They are stable compounds and are resistant to degradation. They form a waterproof layer cuticle on the surfaces of some plant leaves, roots, and in this way reduce the rate of water loss. Water barrier waxy layer generally covers bodies of some animals such as sheep and insects. Steroids. Steroids are lipids that do not contain fatty acids. Each steroid is formed of backbone of four fused carbon rings containing 17 carbon atoms. They differ from one another by type of functional group attached. Different steroids have important functions in the bodies of humans and other animals. Cholesterol is a representative example of the steroids. It plays a role as an essential component in animal cell membrane. Cholesterol is also a precursor of all steroid hormones such as aldosterone, sex hormones, and vitamin D. Aldosterone helps to regulate the sodium content of blood. Sex hormones help to maintain male and female characteristics. Terpenoids. Terpenoids are lipids that, like the steroid, do not contain fatty acids. They are lipid soluble and water insoluble substances. Terpenoids are formed of units called isophenolic units. They join by the process of condensation and give rise to different types of compounds such as rubber, carotenoids, etc. Long-term energy storage molecules in plants and animals. Plants usually store lipids and seeds as oils, unsaturated fatty acids, while animals store them as fats, saturated fatty acids. Lipids store greater amount of energy than carbohydrates because they contain less oxygen and more such bonds than carbohydrates. Lipids are building materials of cellular membranes called lipoprotein membranes. Lipids provide insulation to various organs and to the bodies of animals as a whole. Some hormones are lipids that regulate various processes inside the bodies of plants and animals. Example of plant hormones are auxins, gibberellins, cytokinins, while that of animal hormones are aldosterone, testosterone. Nucleic acids. Nucleic acids are polynucleotide chain polymer found by linkages of unit called nucleotides. Here are two types of nucleic acids: deoxyribonucleic acid and ribonucleic acid. DNA is formed of deoxyribonucleotides, while RNA is formed of ribonucleotides. Nucleotides. In a typical nucleotide, the nitrogenous base is attached to carbon number 1 of pentose sugar, while phosphate group is attached to carbon number 5 of the pentose sugar. The bond formed between phosphoric acid H3PO4 and hydroxyl group of pentose sugar is called ester linkage. In a sugar, in a polynucleotide chain, one phosphoric acid is attached to the OH group of carbon number 3 of pentose sugar, while another phosphoric acid is attached to OH group of carbon number 5 of pentose sugar. This is called phosphodiester linkage. Nitrogenous bases are classified into two main types: pyrimidines and purines. Pyrimidines are single ring compounds, while purines are double ring compounds. Pyrimidines are further classified into three types: thymine, cytosine, and uracil. Purines are classified into two types: adenine and guanine. Nucleotides are classified into three types. Here are mononucleotides, dinucleotides, and polynucleotides. Mononucleotides. As the name indicates, a mononucleotide exists as a single nucleotide. One of the common mononucleotide is the adenosine triphosphate (ATP). It is a nucleotide with three phosphate adenine base linked to pentose sugar. Ribose forms a structure called adenosine. When adenosine is bonded to a single phosphate, it will form a nucleotide called adenosine monophosphate. This reaction needs energy. Adenosine monophosphate is a nucleotide with one phosphate group. Another phosphate is bonded to this molecule. When energy is supplied, it forms adenosine diphosphate. With the addition of a third phosphate and energy to it, adenosine triphosphate molecule is formed. Adenosine triphosphate is an energy-rich molecule and is commonly called energy currency. It provides energy to cell of all living organisms for their functions. The very bond between the phosphate groups indicate high energy bond. Usually, the bond between the second and third phosphate breaks up, releasing the energy used by cell. An ATP molecule is hydrolyzed into ADP and P, and almost 7 kcal energy is produced. ATP molecules are produced during respiration when food molecules are broken down into simpler substances and the stored chemical energy is released. This energy is used in generation of ATP molecules. Dinucleotides. When two nucleotides are linked together, a structure called dinucleotide is formed. If the dinucleotide contains adenine as nitrogenous base, then it will be called adenine dinucleotide. Nicotinamide is a vitamin called nicotinic acid. Niacin, thymine is also vitamin called riboflavin, vitamin B2. The adenine dinucleotide in combination with different vitamins form important compound called coenzymes. The important coenzymes are NAD, nicotinamide adenine dinucleotide, NADP, nicotinamide adenine dinucleotide phosphate, and FAD, flavin adenine dinucleotide. These coenzymes can exist in two forms: a reduced and oxidized form. In the oxidized state, they function as hydrogen acceptor. NAD and FAD are both an electron transport chain of respiration where they act as electron carriers. In the process, they are alternately reduced and oxidized. Polynucleotides. When many nucleotides are linked together, they form a structure called polynucleotide. DNA and RNA molecules are polynucleotide chains. There are different types of RNA molecule. Each type performs specific function under the instruction of DNA. Nucleic acid are two types. DNA, deoxyribonucleic acid made of deoxyribonucleotides. RNA, ribonucleic acid made of ribonucleotides. DNA. In 1962, James Watson, 1928, Francis Crick, 1916 to 2004, and Maurice Wilkins, 1916 to 2004, jointly received the Nobel Prize in Physiology of Medicine for their 1953 determination of the structure of deoxyribonucleic acid DNA. According to this model, a double helix. The double helix can be visualized as spiral staircase wound around a central axis. Watson and Crick suggested that bases were always consist of theorem pointing toward pyrimidines, keeping the molecule diameter at a constant 2 nanometer. The base pair are flat with distance of 0.34 nanometer between them. DNA contains pentose sugar as deoxyribose. It is formed of four different types of nucleotides. These nucleotides are named after the base present in them. They are adenine deoxyribonucleotide, guanine deoxyribonucleotide, thymine deoxyribonucleotide, and cytosine deoxyribonucleotide. These four types of nucleotides are used as of DNA molecules. The nucleotides and DNA molecule are bonded to one another in such manner that the sugar of the nucleotide is linked to the phosphate group of the next one. In this way, the nucleotides form a linear molecule called a strand in which the backbone is made of sugar alternating with the phosphate group. The bases are projected to one side of the strand. The sequence of the nucleotide in one type of DNA as constant while it is different from other type of DNA molecule. DNA molecule consists of two strands. The two strands twist about one another. The form of double helix. The two strands run in opposite direction to each other. In the double helix, they are held together by hydrogen bond between furan and pyrimidine bases. Thymine is one strand is always paired with adenine in the opposite strand, and guanine is always paired with cytosine. There are two hydrogen bonds between adenine and three hydrogen bonds between guanine and cytosine.